Good morning, everyone. My name is Andrew Milner. I'm a fourth-year graduate student at the University of Mississippi Medical Center under the guidance of Dr. Michael Garrett. The title for my talk today is From Genes to Proteins, Exploring Nephron Deficiency in the HSRA rat model. So at first, I'm going to cover some background on the model, go over the study objective, the experimental designs for both the single-cell RNA-seq and the discovery proteomics. Then I'll get into some of the analysis and comparisons. Then I'll talk about conclusions and future directions. So my project involves studying the kidney. These are two organs. Their main job is to filter blood and produce urine. They do this through the microscopic unit of filtration called the nephron. So here's the nephron. You've got the glomerulus where the blood is filtered and the filtrate enters the tubule, and you have reabsorption of about 99% of the filtrate. And what's left afterwards is excreted as urine. And so this number of nephrons varies greatly from individual to individual. And it's actually the number of nephrons is set at birth. In 1988, Brenner et al. Put forth the hypothesis that the number of nephrons is inversely correlated with the risk to develop hypertension and chronic kidney disease. And numerous animal and clinical studies have proven this hypothesis. So in order to study the nephron deficit, our lab developed a novel model from the heterogeneous stock, which are comes from 8 inbred progenitor strains. One of them exhibits unilateral renal agenesis at 10 to 15% of the time. And so this results in the heterogeneous stock exhibiting unilateral renal agenesis at about 1 to 2% of the time. It was noticed that one of these families within the heterogeneous stock had increased rates of unilateral renal agenesis at about 50%. And so subsequent inbreeding of this family resulted in the HSRA rat, or heterogeneous stock-derived model of unilateral renal agenesis. And so it has unilateral renal agenesis 50 to 75% of the time. But more importantly for my project, it has altered nephrogenesis in that remaining kidney. So in the single kidney offspring, they have about 20% less nephrons per kidney when compared to 2 kidney littermates. So within this population, we've got 2 groups, the HSRA-C, or 2 kidney offspring, and HSRA-S, or the 1 kidney offspring. So the study objective was to determine if there are early molecular differences in the kidney between 1 kidney and 2 kidney animals. So in order to look at this, we performed single nuclei RNA sequencing and also discovery proteomics. And so that's what I'm going to share with you today. But overall, my project is going to involve looking at transcriptome, proteome, and epigenomic differences. And while I'm not directly involved in it, this should directly complement the ongoing QTL analysis, which seeks to identify genetic loci associated with the number of nephrons. So while I'm not directly involved with this work, I know where I'm at, so I'm going to show a little bit of that data. And so we cross the HSRA rat with the ACI rat, which also exhibits the unilateral renal agenesis, and then estimated nephron number using an acid maceration technique. And so it's physical counting of glomeruli. And then through genotyping, there was an association on chromosome 4 for a loci that's associated with the nephron number. And so the goal here is that my work should hopefully complement this work. So now on to what I'm working on. For the single nuclei RNA sequencing, we took kidneys from 3 HSRA-S and 3 HSRA-C rats, all male. The library was prepared using the 10x Chromium controller, and it was sequenced on the NextSeq 2000. I then performed bioinformatic analysis using Seurat, and I was able to identify clusters of kidney-specific cell types. And so this plot on the left is a UMAP plot where each dot represents a single cell, and its location in the plot tells you about its relatedness and gene expression to other cells. And so cells that cluster close together are more related and can be denoted a certain cell type. And so these were manually annotated using canonical markers of kidney cell types. And so the violin plots for some of those markers are on the right. The next step is differential expression analysis. So we can separate out the 1 kidney and the 2 kidney groups. So that's on the left, you've got all the cells in the 1 kidney, and on the right, all the cells in the 2 kidney from both groups. And so this would be more comparable to like bulk RNA sequencing. So overall, we found 160 differentially expressed genes, and then filtering by a 50% fold change, there were 6 of them. And those are those genes there. I included this just to show you that with single nuclei or single cell RNA sequencing, we can separate out clusters or subpopulations or cell types and just compare the gene expression between those cells. And so here was, it's slightly different between the podocytes than it was globally. And for sake of time, I won't get into every single cluster. When I saw the highly fold change differentially expressed genes, I did a literature search because I was interested if any of them have previously been linked to kidney function or development. And so they actually have been. Differential methylation at FKBP5 is linked to CKD, or chronic kidney disease. ZBTB16 is a transcription factor that's heavily involved in stem cell differentiation during development. P3H2 has been linked to glomerular basement membrane regulation in mice. And then PLZF, which is another name for ZBTB16, it's been shown to inhibit transcription of c-KIT, which this was really interesting because it's been shown that one of the progenitor strains, the ACI rat, the causative factor is a long terminal repeat in intron 1 of the c-KIT gene. And we've shown that the HSRA rat shares this causative element, but that doesn't explain the increased penetrance of unilateral renal agenesis. And then the last one, leukemia inhibitory factor receptor. In a human population, missense variants have been linked to renal hypodysplasia and other forms of congenital anomalies of the kidney. So on to the discovery proteomics. This was performed through the IDEA National Resource for Quantitative Proteomics at the University of Arkansas. So this is the protocol that they followed: tryptic digestion of the peptides, chromatography, and then mass spectrometry. And so we did a larger cohort for discovery proteomics. Again, all male with 9 HSRAS and 12 HSRAC rats. This was less interesting. So overall, at an adjusted p-value, we had 4 differentially expressed proteins. And one of them was PODXL, which is a protein expressed by podocytes, which makes up the filtration barrier in the glomerulus. So we see a downregulation of that protein in the 1 kidney offspring. But at an unadjusted p-value, we see 366 differentially expressed proteins, and then further filtering this by fold change at 100% gives 23. And this is the list there. So in order to compare the single cell or the single nuclei RNA-seq data and the discovery proteomics, I performed pseudo-bulk RNA sequencing, which basically get rid of the cell level metadata and aggregate all the gene counts to the sample replicate level. And then this allows you to use more robust existing differential expression frameworks like DE-seq2. And so the reason that I performed this is that single cell data is known to have really inflated p-values because each cell is treated as a single sample. And also highly expressed genes are shown as differentially expressed and it has low sensitivity for genes that have low expression. So I did a simple Venn diagram to compare the Seurat analysis, the DE-seq2 pseudo-bulk analysis, and then the protein differential analysis. And so we saw a decent correlation between the Seurat and DE-seq2 analysis of the most significant genes, but overall it did not correlate with the discovery proteomics. And so in conclusion, the identification of these differentially expressed genes suggests that transcriptional alterations might play a role in the nephron deficit observed in the HSRAS rats. And then the proteomic analysis highlighting 23 proteins with pronounced variations. These proteins, if functionally characterized, might offer insights into the mechanisms that contribute to the phenotype differences. And then the discordance between these 2 data sets underscores the multi-layered complexity of gene expression and its regulation in biological systems. So this gets me thinking of like post-transcriptional modifications, differential rates of protein degradations, or translation efficiencies. And so like I mentioned earlier, the ongoing QTL and eQTL analysis, it's hopeful that the differentially expressed genes and differentially expressed proteins could be causatively linked to this nephron number. And then in the future, we're working on that epigenetic investigation, reduced representation by sulfite sequencing and small RNA sequencing, and then also single nuclei RNA sequencing of fetal kidneys during the developmental stages. So this is gestational day 15.5 and 17.5. Because like I said, at least in humans, the nephrons are developed by birth and set. I think in rats, they kind of continue to develop afterwards. And then also, we mentioned some in vitro models earlier. The plan is to use the kidney organoids, hopefully as an in vitro model of kidney development, like a molecular testing grounds to test some of these future candidate genes. These are my references for the articles that I mentioned earlier. And I'd like to acknowledge the Department of Pharmacology for their support, Dr. Garrett and everyone in his lab for all the training and help, UMMC Molecular and Genomics Core for all the sequencing, and then the IDEA National Resource for Proteomics at the University of Arkansas, and also all of our funding sources. I'd like to thank you guys for being here and listening, and I'll take questions.